In this lesson, we'll consider what scientists call the reactivation hypothesis. The idea that brain regions used during perception may also be involved in storing memories of those perceptions. Now, in the previous lesson on the working memory system, we said that working memory can include contents such as verbal thoughts and visual imagery, and these are internally generated stimuli. So a thought might just be that voice we hear in our head, or we can imagine something. We can close our eyes and have an image of something in our mind's eye. And we said that for imagery, for example, the visual systems were being activated, providing the input to the working memory system. Well, what's the evidence for that story? We'll look at some fMRI data, and we've already seen Canwisher's studies on the fusiform face area. She identified a small region of brain tissue that seemed to be selectively active for faces compared to other objects. Here we see the fMRI signal here for a face, and then reduced signal for an object, increased response for a face, etc. Now this region of brain is on the underside of the brain, on both sides, but more pronounced in the right hemisphere. So when we think about face perception, then we're going to say this is our fusiform face area here on the right side, but it's really sort of tucked underneath the temporal lobe there. The face area becomes active when we are currently perceiving a face. So eyes open, looking at a face, electrical signals are sent to visual cortex and then to the face area, and we get that selective response in the fusiform face area. So the idea is then that this face area is providing the contents, in a sense, to working memory. So we consciously perceive a face. Now, it turns out there are other areas of the brain that selectively respond to stimuli as well. So in blue, we have the face area. Notice it's a little bigger on the right side, so we're looking at the underside of the brain. And then in pink here, we have two regions here, also in the left and the right hemisphere, what scientists call the parahippocampal place area. These are brain regions that respond to scenes of outdoors or houses, so nature scenes or houses, that kind of thing. So we'll call this the place area. It's important to note, however, though, that the place area and the face area do not overlap. So scientists can distinguish between activity in the face area in blue and the place area in pink. And that'll be important for the following study. Like in earlier research, Canwisher began by putting subjects in the fMRI machine and then showing them a series of images on a computer while recording the activity in the fMRI machine of the face area and the place area, those distinct regions of the brain. The face area has a preferred stimulus of a face. The place area has a preferred stimulus of some scene, a house or outdoor scene or something like that. So here we go. When the subject sees a face, we get a significant increase in signal in the face area, not much significant increase in the place area. But when the stimulus switches to the house, then you have a reduction in activity in the face area and a significant increase in the place area, just as you would predict. These two regions of the brain are selectively responding to their preferred stimuli. So what we have then here is you can just sort of go back from one stimulus to another. Faces activates the face area, then the house comes along and the place area gets activated. And so you see this shifting in local brain activity in the two places, in the two regions of the brain, depending on the stimuli that is presented to the subject. Now here's the interesting part. Canwisher then gave instructions to the subjects to simply imagine a face or a place, right? A face or a house. When they imagine a face, you get fusiform face area activation. It's not as much as seeing a face, but nevertheless there is a reliable signal here. Notice in the place area we don't have any increase in signal when the person's looking at a face. But then if they imagine a house, the activity in the face area goes down, activity in the place area goes up. And so again we get that same kind of pattern that we saw for perception. We see that again in imagery. Now think of what it means to imagine a face, right? We can close our eyes, no external stimulation, there's no face that we're seeing, but we're simply internally generating the image of a face. So in a sense, we're reactivating a memory of what a face looks like, or maybe somebody we know, and likewise with a house. So imagery involves, reactivating stored memories of the stimuli that we're imagining. And notice we're getting activity in the face area for imagining a face, and we're getting activity in the place area for imagining a house. So what this is suggesting then is that during imagery, when we have to reactivate memories to generate the contents of our imagination, we're getting activity in the same areas that are used for the perception of those same kind of stimuli. So the fact that face perception regions are active for imagery suggests that the face area is used in the reconstruction of a face memory or participates in storing the face memories. So again, the idea is that during imagination, we're not using our perceptual system, so the visual cortex is not being stimulated by the outside world. Rather, we're sending a signal internally to reactivate the face area to generate an image of a face in our mind's eye. And this suggests then that the stored memories of faces may involve some of the same brain regions, the face area, that is involved in the perception of faces. We're going to call this the reactivation hypothesis. The reactivation hypothesis is the idea that those regions used to process stimuli are also used to store memories that involve those stimuli. And we might also note here, recall from the fMRI data, that the activation of the face area was reduced compared to perception. So there's activity in the face area when we imagine a face, but it's not as much as when we perceive a face. And that may simply be due to the fact that when we're imagining, we're not activating that visual pathway. So the face area is not getting that normal kind of perceptual activity from the visual pathway during perception. So this may explain why it is that the signal for imagining a face is smaller than the signal for perceiving a face. The face area is just not getting as much neural activation because during imagery, we don't have our eyes open, we're not seeing a face. We're internally generating an imaginary face by activating a memory of a face. Now, if the reactivation hypothesis is true, then it makes certain predictions about what should happen when a subject suffers brain damage. Let's take a look at three regions of the brain that seem to specialize in different visual processing. There's a region of the brain that becomes active when subjects see objects that have color. So in other words, that this region seems to be processing color. And in fact, when it's damaged, people report blindness, color blindness. Now they can still see objects and things, but there's no color to the objects. A little farther down in the visual pathway, damage here interferes with the ability to recognize objects. So this is the area of the brain that is sort of processing object shape that we use to recognize objects. Damage here interferes with perceiving objects. And then we have the face area here. When that's lesioned, people have difficulty recognizing faces, and that's called prosopagnosia. Now here's the important point. If the reactivation hypothesis is true, and these bits of brain tissue are involved in perceiving some visual aspect of the world, but they're also involved in storing memories for those aspects, then damage should interfere with both perceptions and imagery and memory. And in fact, people who have damage to this region of the brain, they report that they can't see colors. They also cannot imagine colors. So we have the deficit in the memory, in the imagery. And likewise, with damage to this region of the brain, people have trouble recognizing objects. They also have difficulty imagining objects. And likewise with faces, difficulty perceiving faces, also difficulty imagining faces. So the lesion data supports the reactivation hypothesis. Lesions interfere with perception, but also are interfering with imagery, which is an aspect of memory. There's another line of evidence that gives some support to the reactivation hypothesis. This is the early work of Penfield in his stimulation studies. So when he was stimulating visual areas of people who were awake and conscious during brain surgery, stimulation of the visual areas produced visual memories. whereas stimulation of the auditory cortex and surrounding regions produced auditory memories. And again, this fits with the reactivation idea that the visual areas are used for visual processing during perception, but stimulating those same areas can produce visual memories, arguing that the same bit of brain tissue is processing both perception and may be involved in storing memories, in this case visual here and auditory around the auditory cortex. So in summary then, the reactivation hypothesis asserts that the same regions of brain that are used for perception, in this case faces, the fusiform face area, that region of brain is active for perceiving faces. It's also reactivated when we're imagining a face. Now, imagery is, in a sense, a reactivation of internally stored memories, so the suggestion is that the regions used for perceiving faces are somehow involved in storing the memories of faces as well.